We're okay, right?
Yeah.
Okay. Good morning, everyone. Welcome back to another fine weekly seminar. Like we normally do, we'll just take a couple minutes to briefly share with you what we accomplished last week. And very quickly, let me close that. And very quickly mention next week. So last week we heard from Aubrey Kelly, who is in the Department of Psychology at Emory University in the US, and she gave a wonderful presentation describing her research on the neural mechanisms underlying grouping behavior in spiny mice. So very, very interesting talk, learning again about a new system where people are trying to do proximate mechanisms of social behavior. Before we go on to today's talk, next week we have one of those somewhat different meetings scheduled, a fantastic panel on animal behavior and conservation. We're going to be joined by 4 colleagues: Odit Berger-Tal from the Ben Gurion University in Israel, Dan Blumstein, who has already presented at FINE. He's at the University of California in LA. Alison Gregor at the San Diego Zoo Wildlife Alliance in the US, and John Swaddle, who is in William and Mary College, also in the US. Like we have done, though, those of you who may not have attended other panels, what we're doing, our students, the members of our teams are helping us put together a number of questions that we will send to the panelists very soon, so that all 4 of them address those questions to begin with. So we will hear from them for about 40-50 minutes, like we've done in other panels. And after that, like it is defined culture, we'll open the meeting to hearing from everyone. So you will again have a chance to pose questions to the panelists, and I'm sure we'll have a wonderful conversation going on for a full hour. This is next Tuesday, October 24th. With that, I'm going to stop sharing my screen and introduce my long-term friend and colleague, Ben Finkel. Ben, I first met Ben, I don't know when I first met him, but the year that comes to my mind is 2013. So it's been 10 years that we have been talking, communicating. Ben went to Argentina to work in the Almaty project for about 6 months. Then he came to Yale, where he stayed for a year, helping me get on many, many things. One of them was what ended up in some huge pieces of writing we did with Tony and Sari. We're missing Macken, I don't know if she's there. Where we really looked at data for pair bonding and the kinds of topics we study. Ben went on to study his PhD working with John Mitani at Michigan University in the Gogo community of chimps in Uganda. And he tells me that maybe, if he had talked in a few months, we would be celebrating his completion of the PhD. For all purposes, it's done. We just need, he's waiting for some very clerical administrative steps. So for all purposes, we can call him what he is now. He's a visiting assistant professor at Boston University. He's been there for a few months. Before that, in terms of the research he's done, and he'll be presenting some of that, he's got a Leakey Foundation grant that helping support the research in Uganda. He's also got an NSF grant. And from what we were discussing today early in the morning, there are many, many papers that I know are going to be very, very exciting that he's working on that are derived from his research. So it's somewhat of a privilege. We're going to be hearing firsthand about this research that he's putting together and submitting for publication in the near future. Another angle that's important to us in FINE, and I know this because, again, it's been 10 years talking to Ben. He's a fantastic educator, wonderful speaker. He's always, we have shared for a decade a common interest in how to raise the excitement in young students. So he's done a lot of teaching. He likes that. He's teaching right now. A lot of outreach work as well. So with that, I want to leave the floor to Ben, who's going to be discussing the benefits, senescence in male chimps. So Ben, if you want to take it from here, just one more word. Remember that he'll be talking for the first hour, in case some of you need to go an hour from now at noon Eastern time. And after that, as it is customary in FINE, we will take an hour 45 minutes because he has many individual meetings coming up. We'll finish today at 12:45. We'll take questions from you via the chatting window. With that, Ben, welcome, and please take it from here.
Eduardo, thank you so much for that incredibly generous introduction. I truly hope I can live up to it, and I am simply honored to be here today and get to share my research with you, and hopefully an appreciation for these really singular apes. And I'm going to go ahead and start sharing. We had a little bit of technical difficulty, so I am just going to try one method, and if that does not work, we are going to our PDFs as our backup.
So
Eduardo, would you mind?
So far, so good. We have it. So far, keeps going. Yep, that's good. That's good. Yep.
All right.
It's working.
Okay. Let's see. Apologies. Just need to get my presenter notes up. Okay. Hello, everybody. And again, my name is Ben Finkel, and I'm a member of the faculty at Boston University. I'm also a biological anthropologist and primatologist. And as you see today, I also dabble in evolutionary ecology. And today I'm really excited to share with you some recently completed research that's now being worked up for publication. And consequently, I just ask that folks not tweet or share preliminary figures and results. Thank you. So here is a chimpanzee we call brownface eating leaves from a Terragotta sapling. He is the Ngogo community's oldest male here at 53 years. And brownface's age is apparent in several ways. For instance, he has only 8 remaining teeth, most of them nubs. Yet brownface retains much of the normalcy of adult male life for a chimpanzee. He climbs trees to forage, he travels with the group, he even hunts monkeys enthusiastically, I might add. And so similar observations on the robustness of old age chimpanzees has led to the conclusion that our closest relatives, like other long-lived animals, manage to age gracefully. So my question is, what does aging actually mean for animals like brownface? And what can this tell us about the evolution of longevity? And since 2016, I've had the privilege of working with the largest, most long-lived population of chimpanzees at Ngogo in Uganda. And this work today comes from my recently completed dissertation work at the University of Michigan. And based on this question, and today I'll be exploring that and going along with a fine core theme of how and why animals live in different societies. Today, we'll be getting a chance to explore the consequences of these societies on animal well-being and lifespan. So in the next about 45 minutes, I'm going to attempt to walk you through.
Ben, sorry,
yeah.
It's broken up. It's like it
is broken up.
No, okay, it takes, now it's fine. It is a little bit slow in loading the image. Now it looks good. Let's see what happens. Keep going. Sorry.
Okay. No, please interrupt if it's not working. Just let me know and we'll adjust. Okay, so I'm going to give you an overview of relevant background and introduce my study, design, and methods, and then I'm going to present findings from 3 separate investigations attempting to tie them together. First, the foraging on investigations into foraging performance and aging, how older chimpanzees may balance trade-offs, and the consequences of these for social aging patterns. So senescence comprises the set of age-related changes that adversely affect an organism's vitality and function. And this multitude of deteriorations that accumulate with age present what we see as aging bodies and minds. And importantly, as we know, senescence is evolved. To summarize, late life reproduction is more visible to natural selection. And late life
Ben, I'm sorry, but I think
no, that's okay.
It is there. Now it goes again. Can I get some more people commenting on whether is it also slow to most of you that it's taking a few
slow,
seconds?
But it comes eventually. It's slow, but eventually it comes. So
Yeah.
So my suggestion then, if it'll give you an experience, an unforgettable experience that you have to change as you go. Give it a few seconds. I mean, just take a deep breath, and we'll bear with you when you click the next slide, so that you don't start talking before we're actually seeing the text. That would be my fix.
Yes,
Like,
I will. I'll try to
on.
anticipate the delay. All right. So senescence is a result of trade-offs between early and late life fitness, and harmful traits are allowed to accumulate over the lifespan because of the selection shadow. So to understand the evolution of longevity, we must know how animals maintain both reproduction and survival late in life. Senescence was once considered rare or even non-existent in wild animals, simply because most were expected to die before they could get old enough to senesce. But thanks to an increasing number of long-term studies, we now know that senescence decreases fitness and survival across the animal kingdom, particularly vertebrates. And most of these studies have been done on birds and mammals. Despite this increasing knowledge of senescence in wild animals, we still know little about the mechanisms through which aging impacts their fitness. This figure illustrates the progression of biological aging across levels of analysis, from the proximate accumulation of molecular damage to ultimate outcomes in reproduction and survival. So most research takes place at the far right in the wild, which is also known as demographic aging, the bird's eye view that tells us how much survival and reproduction may decline with age. However, a more complete picture of senescence will identify the nature of functional decline, a mechanism that links senescence and fitness. For instance, recent findings in particular have highlighted how functional declines such as immunosenescence, and how these may lead to increased mortality in wild animals. Noah Snyder-Mackler gave an excellent prior Fine talk and has published on this. Even to those who do not study wild animals, I recognize this may seem a little obvious. For instance, it's self-evident to those with aging pets that older animals have fewer abilities and greater needs. However, the experience of wild animals are quite different from our pets, and so is their study. Wild animals, including chimpanzees like brownface, must persistently sustain themselves, meeting daily dietary requirements and avoiding threats. So to understand the evolution and ecology of senescence, the study of organisms under natural conditions is critical, because this is where natural selection operates and functional declines can translate directly into loss of fitness. And in particular, there have been relatively few explicit connections between functional and demographic declines as mediated by behavior in studies of wild animals. With that, let us turn to wild chimpanzees. Chimpanzees are important models for studying aging because they are especially long-lived, and as an anthropologist, because of their close evolutionary relatedness to humans. Survivorship curves, such as the one that is hopefully coming up on your screen now, tell us that how many chimpanzees are expected to survive to a given age. And so you see that each line here depicts a different study community. And you can see there's a tremendous amount of variation in how long chimpanzees live. At birth, the chimpanzee at Tai in the Ivory Coast has a mean life expectancy about 13 years, whereas one at Ngogo has a 33 life expectancy in this study. So chimpanzee life history has been most closely studied to compare humans. And here, Wood et al. Found that the Ngogo chimpanzees seem to have more similar survivorship curves, even to that of human hunter-gatherers, compared to other chimpanzee groups. So today I'll be presenting findings from this population, from this community, in hopes that they can provide particular insights into patterns of senescence in the wild. Furthermore, I'm going to present findings today on male chimpanzees. As in many mammals, males have lower life expectancy than females, living about 6 years shorter on average at Ngogo. At Ngogo, we may say that prime adulthood in males begins at age 21, whereas there's some evidence that individuals begin to exhibit certain declines after age 30. Because chimpanzee males sire offspring into old age, selection pressures in this demographic group are particularly relevant for individual fitness, and perhaps more so in most species, or rather in species where individuals are not reproducing late in life. Consequently, natural selection in past prime males is likely to have evolutionary relevance for chimpanzees. And in other words, older males may still be visible under the natural selection shadow of aging, or sorry, the selection shadow of aging. So this is one explanation for the relevance of aging in males, but I will conclude with a few notes on what I hope to learn from future studies of female chimpanzees. By many measures, chimpanzees appear to be good at aging, maintaining condition right up until their final days. We see that survivorship does not dramatically decrease with age. Meanwhile, some studies show that older chimps don't spend their days much differently than their prime age counterparts. And Dr. Emily Otala, a Ugandan researcher and conservationist, summed this up well, saying, "They handle old age much better than we do. They just power along. It's amazing." Yet we know that aging males do experience declines in fertility, increased muscle loss, high rates of viruses and immune activation, as well as increased stress as measured by glucocorticoids. And taken together, this presents a puzzle for how senescence actually affects male chimpanzees. That led me to my research question. And hopefully, I know sometimes there's lots of animations, and so they may be a little delayed. Thank you for bearing with me. So my question is, what are the challenges of senescence for male chimpanzees, and how do they meet them? Specifically, I will try to identify an area of functional decline, asking whether foraging performance declines with age. Second, asking whether aging chimpanzees face shifting trade-offs, and do they rebalance in order to age gracefully? And lastly, and perhaps of most interest to this group, of how do these challenges relate to known and newly observed patterns of social aging? With that, I'd like to briefly describe my study design. Ngogo, I've been talking about it a lot, but it is located in Kibale National Park, Uganda, under the Ngogo Chimpanzee Project. And so shown here in relation to other chimpanzee sites in the same park. And importantly, these are known individual chimps habituated to human researchers. And so since 2016, Ngogo has consisted of actually two separate communities or chimpanzee groups, referred to here as Ngogo Central and Ngogo West. And as I've mentioned, Ngogo is particularly long-lived. And one explanation for this phenomenon, and an important characteristic of the site, is that it has abundant food, particularly ripe fruit. And this shapes the local behavioral ecology, even in contrast to the nearby chimpanzee groups like Kaniwara, lying less than about 10 kilometers away. So I collected cross-sectional data on 20 adult males over 1 year, along with my research assistant and now collaborator, Sharifa Namaganda. Here's a look at my study subjects, which included 10 individuals over the age of 30. And from these observations, we completed nearly 1,300 hours of focal observation. And we also collected fecal and urine samples. And I will be presenting results today from this larger data set. And I will be providing specific methods relevant to each study as they come up. Here is a simplified representation of my analytical approach for building how I build models to understand the effects of age. So I'm interested in age's effect on a suite of behavioral and physiological outcomes. So such measures here are labeled as outcome, and I considered alternatively linear effect of age or a quadratic effect of age squared because of documented nonlinear effects of age. But of course, we know that many things affect chimpanzee behaviors, from cyclical cycles of seasonality to time of day to dominance rank. And I'm not going to go into detail here, but I employed established methods for generating covariates such as rank and party composition. So because we're not certain about these predictors relative approach for a given analysis, I built a wide series of models of this sort, generalized linear mixed models, selecting appropriate distributions for non-normal data, and I compared their performance in a principal way using AIC or corrected Akeke's information criterion. And the results you're going to be seeing today are from model averages, which permits me to incorporate model uncertainty and insights from various sets of models. And this is pretty standard state-of-the-art statistical methods within anthropology. I did employ a few novel techniques to generate certain predictors like diet quality, which I'd be happy to talk more about. Now for the first investigation on the nature of functional declines for male chimpanzees. Foraging performance may be a particularly important link between cellular and demographic aging because it's dependent on body condition and in some cases is a determinant of individual fitness. Foraging is the set of processes that organisms use to acquire nutrients, and foraging senescence, simply put, should make it harder for old chimpanzees to get food, or old individuals of any species. So evidence has begun to emerge for foraging senescence across taxa, from walruses to bees to albatross. So for instance, based on studies of long-lived albatross, Lecomte et al. Proposed that foraging performance may be one of the first phenotypic traits to reflect aging in natural conditions, referring to it as a potential cornerstone of aging. And so while evidence of how widespread this phenomena is mixed, there's some evidence that feeding performance may decline with age in primates. So the aims of this research is to illuminate how an organism's aging physiology may impact nutrient acquisition, and specifically, I test my hypothesis that foraging performance declines with age. So Stuart Altman wrote that in the course of evolution, every organ system of animals has been altered by the exigencies of obtaining food. Foraging is multifaceted, so how can we measure it? What exactly constitutes foraging performance? Of course, there's no single measure. So here I present a conceptual framework to break foraging down into 3 primary goals and each of their respective actions. So the gray boxes represent activities and the interior white boxes represent my measurements. So first, animals access food by locomoting, and in the case of chimpanzees, often climbing. They consume food via selection and ingestion. And animals process food by chewing and internally breaking down matter. And I'll be presenting my measures for each of these outcomes momentarily. So because senescence is a systemic deterioration that shows synchrony across somatic systems, I predicted that the performance of each of these actions will decline with age. For instance, I predicted that ingestion rates across food items would decline as they eat more slowly, as older chimpanzees eat more slowly. So first, let's examine that ingestion rate. To collect data, I essentially counted units of food consumed to calculate units of food eaten per minute, 1 leaf for 1 fruit. And in total, I showed data here from 13.5 hours of ingestion rates across 9 of the most commonly consumed food items. So what was the effect of age on intake rate? To illustrate this, I present weighted model average effects in the coefficient plot. And I'm going to be showing a number of these, so I want to take time to explain what's going on in these plots. So the dot represents the effect estimate for each respective predictor, surrounded by confidence intervals. Those to the right of the gray dashed line of null effect indicates positive effect, increasing a predictor that increases or is associated with increased intake rates, and the left indicates a negative effect. So for those whose confidence intervals do not overlap with 0, we say it has a reliable effect. So the x-axis represents odds ratios or multiplicative values. Here, for instance, a 0.5 value is associated with low quality food items. So for instance, unripe fruit or mature leaves. And these, as you can see by the 0.5, approximate 0.5 mean there, this means that basically these low quality items are consumed at half of the rate, 0.5 of the rate of high quality food items. Importantly, all predictors are standardized so that the magnitudes of effect between predictors are comparable. So for visual clarity, coefficient plots don't include various control predictors like time of day, date, or observer, or those with no clear effect. So here we see that age has a consistent negative effect on ingestion rate across these 9 food items, as the effect is reliably to the left of the gray line. Old chimpanzees ate fewer units of food per minute compared to prime-aged individuals. And here's a visualization of this effect, representing the predictions of the weighted model average across the observed ages, depicting the decline in food intake rate with age. To assess processing performance, we measured fecal particle size. In short, you chew your food in order to break it down into smaller pieces for which nutrients are more readily absorbed by the gut. For various animals, fecal particle size is said to be a measure of chewing efficacy, and my collaborator hosted this laboratory work and pioneered many of these methods. So here I'm going to present two related but uncorrelated measures related to fecal particle size. The first, the proportion of large undigested fecal particles, and the weighted mean of fecal particle size. So now I'll show you what those two actually look like, give a little more context. So essentially, large undigested particles are what I removed from the feces in the field, dried and weighed as a proportion of total fecal mass. These are items greater than 4 millimeters, and this is in short how many large items like seeds and husks were found in the feces. Indeed, there is plenty of whole fruit. So second, we employed a cascading sieve method to separate the remaining dry particles by size to be weighed in order to calculate weighted mean of fecal particle size. So essentially, you're separating them out by size, drying them and weighing them and generating a weighted mean of the size to come up with a single value per fecal sample. Indeed, we found that the proportion of large undigested matter in the feces increased reliably with subject age. So once again, you see the effective age reliably to the right of the null effect line, indicating the positive correlation as depicted by the black line on the right. The weighted mean of fecal particle size was also reliably and positively correlated with age, suggesting that older chimpanzees have lower processing performance. And consequently, we might expect them to absorb fewer nutrients from their food. Importantly, you might remember the oldest subject in my study, Brownface, who had essentially no teeth left. He did have quite a bit of leverage in the model, but even when controlling for that or removing Brownface's samples, the effect of age remains. In summary, I did find that rates of consumption decreased with age, and older chimpanzees had larger fecal particle size than their prime-age counterparts. However, I found no relationship between age and various measures of arboreal locomotion in foraging context, nor did chimpanzees select different food items. So I propose that these declines suggest that older chimpanzees may have reduced nutritional yields from the food they eat, which is important fitness consequences for wild animals. So these findings join just a few observations of declines in foraging ability in primates, documented, for instance, strepsirrhines and geladas. However, I think that this is some of the most direct evidence for this and is the first that I'm aware of in non-human apes. So what's actually responsible, what physiological decline or what aspect of senescence might actually account for this? I think that the most obvious culprit is likely dental wear, as suggested by our brownface anecdote. So chewing more slowly and swallowing larger items is actually completely consistent with how elderly humans respond to tooth loss. And so I argue that this represents a potential instance of functional senescence. So foraging senescence may join other functional challenges with age, such as decreasing body weight and decreased immune performance. So let's move on to address how male chimpanzees might balance or adapt to various challenges of aging. We can turn to life history theory for potential explanations. Life history theory accounts for how animals allocate energy to growth, reproduction, or maintenance. Once animals reach adulthood, they stop growing and face trade-offs just between the latter two. So as biologist David Resnick said, life history theory accounts for how natural selection should shape the way organisms parcel their resources into making babies. No organisms can spend a calorie into two locations, so we expect them to spend it wisely. As organisms approach the end of life, their expectation of future offspring declines. And so this changes the costs and benefits of respective investments. So in response, both theoretical and observational studies of wild animals have shown two strategies that can be adapted for these late life, or to put it dramatically, terminal individuals. First is the terminal investment hypothesis, representing an extreme of a last-ditch investment phenomena where an organisms prioritize reproduction at the expense of other traits and even survival late in life. Alternatively, the terminal restraint hypothesis says that especially during dangerous times, it might be important to forego reproduction. And so this is kind of like in times of crisis, prioritizing the self-care necessary to stick around and fight another day. However, empirical observations often fail to document this clear trade-off. So instead, a negative correlation between reproduction and maintenance, as if they were on opposite ends of a seesaw. Studies sometimes show no correlation or even positive correlations between them. Some animals may be high enough quality to be able to keep both ends of their seesaw in the air. One potential explanation for the absence of clear trade-offs are differences in acquisition or resource management. Think about this as if some organisms are so fit they can indeed have it all, for instance, by being more efficient foragers. Variation in the ability of organisms to acquire nutrients can prevent researchers from noticing clear trade-offs. And consequently, it can be informative to include foraging in models of life history allocation. So this is particularly important for, and perhaps relevant for aging chimpanzees, who, as we've noted, experience several changes in their energetic landscape. Potentially investigation one, they may experience foraging declines. They may have increased costs, energetic costs from immune activation, and they experience other metabolic shifts associated with things like decreased muscle mass. So we may expect organisms to adopt different adaptive strategies regarding resource management. So first, in order to compensate for decreased returns or increased costs, old chimpanzees may increase foraging investment, and I call this the terminal acquisition hypothesis. For instance, elderly humans with worn down and missing teeth are known to spend more time chewing overall. It's unknown whether old chimpanzees are doing the same. Another strategy old male chimpanzees may employ is to restrict expenditure, decreasing many costly behaviors. In short, old organisms may engage in strategic restraint as they are likely to encounter energy-related senescence. Living faster, so to speak, can speed up the process of senescence itself and functions like oxidative stress. Individuals can instead reduce their rates of senescence by living slowly. Consequently, we should consider how life history decisions are made at two loci, not just allocation, but upstream resource management. And I use this framework to present these four hypotheses, two at each loci. And I think this is a useful potential framework for investigating shifts in life history trade-offs with age. Importantly, studies, studies frequently look at life history trade-offs between early and late life, considering how senescence may shift trade-offs over the course of adulthood is less well known. So I first test hypotheses at the resource management loci. I develop the following predictions. First, if individuals prioritize acquisition, I expect them to invest more in foraging. Conversely, if male chimpanzees adopt a restriction strategy, then I expect them to reduce foraging activity along with other energetically expensive activities associated with movement, in particular climbing, because climbing is more energetically expensive than walking. And I predict a particular decrease in climbing behavior. To test these predictions, I collected data on the activity and movement of adult males. To assess activity, we performed behavioral scans at 15-minute intervals, and I analyzed states in these scans, whether resting, foraging, moving, or socializing. And at each scan, I also recorded subject height to calculate the change in vertical distance between consecutive scans. In other words, how much are the chimps actually moving up and down? So first, I present results here on activity budget, in which age was a reliable predictor. Age was positively correlated with foraging time, the interaction with party size. So on days with large parties, when the chimpanzees were in larger groups because of their fission fusion social structure, the foraging frequency increased with age, and on days with small parties, foraging frequency decreased with age. So there was not a consistent effect. And however, with age, I did see that age was an important predictor and older chimpanzees rested more and moved overall less frequently than their prime-aged counterparts. So to recap, I found no evidence that foraging declined consistently with age. However, both activity in resting and moving behavior shifted. I did not see the same patterns in relation to climbing behavior. So I'm moving over those results here. So next, I consider the predictions of how the two hypotheses at the allocation loci, reproduction versus maintenance. So here I consider the effect of age on several more measures in addition to the activity measures I just showed. So because testosterone is correlated with muscle mass, reproductive behaviors, and aggression in male chimpanzees, I predicted that testosterone will conversely maintain, increase, or decline with age. So terminal investment is investing in higher testosterone or maintaining testosterone, whereas restraint is decreasing testosterone, predicts decreased testosterone levels. Next, regarding behavioral events, I looked at several key behaviors associated with reproductive effort. And I'll go over these more in a minute, but I essentially associate that behaviors associated with reproductive effort should stay the same or increase under terminal investment and decrease with terminal restraint in relationship to age, as well as copulation rates themselves. So let's turn to urinary biomarkers. And first, I use C-peptide insulin as a proxy for energetic balance. I collected urine in the field through the very technical bag on a stick method. And I conducted this laboratory work at the Comparative Human and Primate Physiology Center at the University of New Mexico with my collaborator, Melissa Emery Thompson, who, especially in COVID, helped make all of this work happen. Now, results. I did not find an effective age on C-peptide concentrations in urine, indicating that energetic status did not vary with age. And I'll note here that when you look at the energetic status across the 20 subjects here in the order of increasing age, you note that there's very little inter-individual variation. And in fact, this was supported by models showing that there are very few predictors. It seems like these chimpanzees do a particularly good job of balancing their energetic status across seasons. So next, let's talk about the costly hormone testosterone, a moderator of aggression, mating, and dominance. In general, the pattern of testosterone in vertebrates is said to prioritize reproduction at the expense of lifespan in males. So that's potentially a very fitting measure to compare my hypotheses. So testosterone data come from 270 urine samples over my study period. And once again, I used a method previously validated in wild chimpanzees with collaborator Miss Emery Thompson at University of New Mexico. Now, results. Testosterone did not decline reliably with age. There was no difference in testosterone between old and young chimpanzees. So here again, we see the 20 subjects. In fact, the only reliable effect besides temporal predictors like time of day, excluded here for visual clarity, was community. Those two different Ngogo communities had very different levels of testosterone, and I am investigating this in a separate study about the social determinants of testosterone that I'd be happy to talk more about later. Now onto those behavioral observations associated with reproductive effort. First, I looked at displays, which are a component of the male dominance repertoire. Chimpanzees often accelerate over short distances accompanied by vocalizations, sometimes swinging branches or buttress drumming, slapping their hands on tree roots to appear intimidating or solicit copulations. And because of this speed, because moving faster is more energetically expensive than moving slowly, these are likely energetically costly behaviors as well. And they are sometimes, but not always, accompanied by contact aggression. So to calculate their frequency, I recorded all instances of displays by my focal subjects across those nearly 1,300 hours. And I also recorded the distance transverse by visually estimating their displacement from the starting to ending location of the display, having distance estimations for a subset of those displays. We also recorded all instances of mating or copulation by these focal subjects, recording a total of 154 instances. This is about equivalent to 2.2 copulations per 10 hours of observation. I found that with age, specifically age squared, I found that with age, display frequency declined. There was a negative correlation. Older chimpanzees displayed substantially less often when controlling for other important factors like rank. Chimpanzees also displayed twice as often in high quality food periods, as indicated by the effect of percent time forage. Display distance also decreased with age. Older male chimpanzees, when they did display, they did so over shorter distances. Now, copulation rates. Older males did copulate less frequency, as mediated by a condition effect related to diet quality. On days in which chimpanzees spent less time foraging, should be associated as a inverse measure of diet quality. Younger chimpanzees copulated more, but old male copulation rates were relatively consistent across all seasons. So let's recap once again. I find various instances of activity, particularly behaviors associated with reproduction, to be negatively associated with age, that older chimpanzees were overall likely expending less energy. These findings are also broadly consistent with the maintenance hypothesis. However, and really importantly, is that testosterone did not decline with age, and in fact, it remained the same. So while old males may be demonstrating some level of activity restraint, they still are likely maintaining their reproductive condition, even when they are investing less in reproductive behaviors. Overall, my findings suggest that aging male chimpanzees may employ strategic restraint to reduce overall activity levels, consistent with a resource management strategy. However, but importantly, foraging behavior did not change reliably with age. Second, the results provide partial support for the hypothesis that old male chimpanzees may prioritize maintaining physiological condition. But an important part here is that even though their testosterone levels weren't declining with age, maintaining these relatively high levels late in life should be relatively more costly, and therefore may represent a relatively higher investment in reproduction. Perhaps it could be that they are maintaining condition, so when fewer opportunities do present themselves, they are able to take advantage of it and gain reproductive benefits. However, because they are copulating absolutely less, there are various questions about what benefits these late life chimps are getting from their high testosterone. So I think that this represents evidence, potential evidence for trade-offs between shifting trade-offs that accompany senescence in wild chimpanzees. And if these observed correlations are consistent with potentially adjusting behavior in the face of changing payoffs in order to extend the health span of these aging primates. Now, my remaining time, I want to talk a little bit about how these challenges of functional declines and potential shifting trade-offs with age may be linked to observed patterns of social aging. So the negative impacts of social isolation seem to be shared with, so broadly across various organisms, we see patterns of decreasing social engagement with age, measured from various measures such as the number of social bonds to network connectivity. We've had presenters at Fine, Lauren Brent and Noah Snyder-Mackler, speaking about this in particular. And this matters because social connectivity is a determinant of reproduction and health in many wild animals, and especially social group living primates. However, fewer investigations have attempted to link the patterns of physical senescence to these patterns of social aging. When we consider the patterns of chimpanzee sociality, chimpanzee social aging, there have been early observations that older males may be particularly more solitary or more often alone, corresponding with some measures of decreased sociality, decreased investment in grooming. However, a number of recent studies have really highlighted that particularly male chimpanzees may be becoming more socially selective and grooming with fewer partners overall, but actually with those partners, they are more social, having greater proximity to particular neighbors, as well as grooming them more, and maybe even engaging in more mutual relationships. However, most of the studies to document this, particularly work by Rosati et al., have really relied more on the psychological rather than the physiological explanations of what might be driving these patterns. So consequently, because a lot of the research on this is new and it's not something that has been studied in a particularly long-lived chimpanzee community, I asked first, what are the social aging patterns of male chimpanzees in this particularly long-lived community? And secondly, do we see any evidence that aging or these challenges of aging may in some way constrain or shape shifts in social behavior with age, particularly declines in sociality with age? So to measure this, I recorded in scan sampling, I recorded chimpanzees within 5 meters to the focal subject as being in proximity. And proximity is an important marker of affiliation in primates. It's a prerequisite for other interactions such as grooming and signals social comfort. And individuals were considered to be socially distanced when there was no other individual in proximity. And I also recorded all grooming behaviors by the by the focal subject, and in total collecting 125 hours of grooming behavior. So with age, I found that overall male chimpanzees, older chimpanzees, were less likely to be socially distanced or have no other individual in proximity compared to their prime-aged counterpart. Additionally, there were more males in proximity to those to older males than to younger males. And I'll be talking about the interaction effect in just a moment. Because I also found that old males were still less social overall, although the overall pattern was increased sociality, they were still less social during poor quality food times. So for instance, with age, fewer adult males were in proximity on low quality days for old compared to young subjects. And they also appeared to groom others more, but exclusively on good food days. So this was a condition-mediated effect by measures of diet quality. Particularly the proportion of high quality ripe fruit in the diet, or the proportion of overall the day of the total amount of the day spent foraging. So these findings may indicate that patterns of increased sociality in old males, particularly with other adult males, may be facilitated by periods of abundant food, which could convey that old males are indeed sensitive to energetic or foraging requirements during worse food times. But importantly, this is another documentation of increased sociality rather than decreased between adult males, consistent with what we see in new research at other field sites. And although I didn't get into it here, I did test other potential constraints, such as mobility or competition avoidance. And I'd be happy to talk more about that later. But I do not see particular evidence that these old males, that the social patterns of aging males may be shaped by constraints on their movement, such as travel distance or climbing, or avoiding highly competitive social situations. And I think it is important. I'd be happy to talk more about the limitations of these studies, and particularly all cross-sectional studies of aging. But it's worth noting here that survivorship bias could potentially explain some of these results. It is, of course, possible that this is a study of exceptional survivors, even though even though over 50% of male chimpanzees at the site reach over the age of 30, and there's not particular reasons to think that these older males have particularly different phenotypes than the younger males. It's still an important limitation of these findings. So now let's wrap up. In my first investigation on challenges of aging, I found that compared to prime-aged chimpanzees, old males consumed more slowly and, based on fecal particle size, got fewer nutrients from the food as they aged. And I propose that this represents foraging senescence, a functional decline, which may help us link senescence to declines in fitness with age. In my second investigation on strategies in the face of aging, I found that compared to prime-age males, old male chimpanzees seem to prioritize maintaining physiological condition at the expense, such as testosterone and energetic status, potentially at the expense of energetically expensive activities such as reproductive effort. And I argue that such behavioral shifts may represent compensatory strategies to help male chimpanzees maintain bodily condition and survivorship rate late in life. Such flexible life history strategies are likely an important source of variation in animal behavior. And lastly, in a more brief examination of social aging, I did find that old males may be are more social overall, and perhaps more sensitive to ecological conditions such as diet quality. And we're at 45 minutes here, but I would just like to point out that I've been talking about the exceptionality of Ngogo as an important benefit of the study. These are particularly long-lived chimps, but it's important to consider how these settings, particularly abundant food resources, may affect these results. And if you go back at a different time, particularly during a low quality food period, we may have seen different patterns. Next steps, I'm excited at a number of future projects, particularly to continue exploring this idea of foraging senescence as a cornerstone. How does it occur in relation to other known patterns of functional decline, such as immunosenescence? And also to identify and create more links with the potential mechanism of dental wear. And to investigate this, I've recently assembled a library of something like 3,500 photos of these known subjects yawning. And fortunately, this is a longitudinal data set, sometimes having photos of the same individuals at regular intervals over as many as 15 years. And I also think it's important to consider female chimpanzees. I don't have any particular a priori reasons to assume that foraging senescence would be sexually dimorphic. And fortunately, we have longitudinal intake rates to potentially investigate these questions in females like we've done in males. And also to use that same longitudinal dental photograph to look at potential wear differences between male and females, potentially as a test of the disposable soma theory of aging. And I'd be happy to talk more about that. So as an anthropologist, of course, one of the main motivations to study aging in a long-lived primate is because of its particular relevance to humans. Longevity is said to be a hallmark of human evolution. And this can sometimes be overlooked, but life history work has demonstrated that rather than being totally unique, humans do age on the primate continuum, albeit at one extreme. So studying how aging works in our closest relatives is important to understand our own patterns of senescence. And evolutionary pathways to longevity particularly focus around energy efficiency. And so I think that studying how energy acquisition might change across the lifespan in our close relatives may be particularly important to understand the role of foraging productivity in humans. And with that, I would like to thank my tremendous and extremely large team at the Ngogo Chimpanzee Project, my committee members and various co-authors, my colleagues, including my new colleagues at Boston University, who have been incredibly helpful in continuing to develop these ideas and my research, and And thank you all for hosting me here. And I would love to talk more about these fantastic apes. And of course, a shout out to my various funding agencies and support, including the Uganda Wildlife Authority, who who permitted me to have access to study chimpanzees in this magnificent forest. Thank you. I would love to take questions.
Thank you, Ben. Thank you very much. We have 4 minutes to the hour, so for those who need to go, I'll just take a minute of our time. This will stop other screen sharing. Ben, I will stop your sharing. To remind everyone that next week we have a panel on animal behavior and conservation biology. So very quickly, I'm not gonna find it here right away, or maybe I will. There we are. So many of us focusing primarily on animal behavior, usually at somewhat in our discussion, make some statement, most of the times unjustified, of how our animal behavior research is going to help conservation. So if you feel like I do, that sometimes I don't even know what I'm talking about, I'm being a little bit exaggerated, you may want to join us next week on Tuesday. We'll have a panel, we'll have opportunities to discuss with our colleagues, how is it that we can build stronger, more productive links and bridges between those of us who have made behavioral ecology, animal behavior the focus of our research, and those who have made maybe a more applied approach to conservation. So please join us next week. With that, we're going to go to take questions from the audience like we always do at FINE. Thank you, Ben, for a wonderful talk. As usual, I ask that you simply type a question mark in the chatting window. If you're a student, postdoc, just type an S as well. We'll give priority, as we always do, to our younger colleagues, early career colleagues, in stating their questions. So please feel free to go ahead with questions if you have them. I have many, but I try not to be the first one if somebody else has a question. Otherwise, I'll just get started myself. Okay, there we go. Then, oh, no, I can wait. We have a question from a student, Alessandra Mascaro. So I'll hold on mine. Alessandra, if you could be so kind of introducing yourself, telling us where you are, your institution, and your full name, and then just go for the question. You're welcome.
Hi, Ben. I don't know if you can hear me.
Yeah, we hear you well.
Welcome. So thank you very much for the nice talk. It was really interesting, and I'm so great because I would have so many questions, but I just selected one. So just to briefly introduce myself, I am currently a PhD student of the University of Osnabrück, and I study illness and wound tending behaviors in chimpanzees. So I spend a lot of time with male chimpanzees, specifically injured. And my question for you is mostly related to individuality. So I saw that you found no impact on party size in aging. And I was wondering if you had a look on party composition instead. I believe, for example, all male chimpanzees may be not selecting priorly the party size, but rather preferring spending their time with other individuals. And this party composition may have also an effect on other behaviors of, for example, display frequency. So if they spend their time with specific individuals compared to others, I expect them to spend time with individuals with whom they feel more, let's say, chill, and they have also less need to display more frequently, but they, at the same time, they can also copulate with the same rate.
Right. Yes, thank you so much, Alessandra. Right, so in the, and tell me if I've interpreted right. So in the social agent component, I did not discuss changes in party size with age. But actually, you might have noticed the early descriptions of aging really focused on solitary individuals, which is a measure of party size, saying they were the only ones around. They were totally around, which is a cool thing chimpanzees can do with their fission fusion dynamic. So I actually did not show results here directly measuring party size, and there are a few reasons for that. I just looked at the proximity when they were when I was observing them, who was around, not who was in the party as a whole. And that's actually because I had very few over the 13 months of data collection. Although I made attempts to follow solitary individuals, I specifically went out looking for solitary individuals. I actually had extremely few observations of individuals being completely alone. And so I didn't actually directly measure, include results here of looking at party size or true solitariness. Instead, I opted for that socially distanced measure. In short, I think that I needed more data and more reliable measures of party size to be able to assess that. I think it's really important. I just don't think that it was the best use of my particular data to answer this. The Rosati et al. Longitudinal study found that older chimpanzees were basically their average party size was smaller. But when there was a really big party, they were more likely to join it. So they were like more social. When there was a big event happening, a big patrol or a big fruit day, it seemed like older males were more likely than not to show up. And so I think that's really interesting. I just don't think my data set was a particularly strong one to address that.
Thank you.
Thank you, Alessandra. We're going to now hear from Sendur Sarwan. If you can please introduce yourself and let us know where you are and what institution you're joining us from. Sendur?
Hello, am I audible?
We can hear you well.
Yes. I'm K. Sendur. I'm a class 12 student from Bangalore, India. So I just have one question. As part of the social aging section, you mentioned senescent males are more selective with their grooming partners based on proximity, right? So is this also observed in their reproductive behavior? That is, are senescent males more selective when choosing on whom to display their reproductive to?
Yes. Who are they displaying to? Who are they copulating with? In general, it seems like older males, because they are lower, so rank declines with age as well. So it's a really important confounding variable. And we do know that lower ranking older males are essentially probably investing not in the super high quality females, the ones that the highest ranking individuals are attempting to monopolize or go on consortship with or something like that. But they may be, I believe that there's some work that these lower ranking individuals may be copulating more, particularly on the edges of the reproductive swelling of the region in which a female is reproductively available. I don't think that there has been particularly work like parsing rank and age on that. So that's something I'm really interested in looking at the future. But I'm in terms of who are these older males differentially aiming reproductive effort at particular females, I think that's a really important question and something I'm looking into. Did that address your question?
Yes, it did. Thank you.
Thank you, Sendur. I don't see another student, so I'll take the chance to ask the question I had. Ben, one of the hypotheses, one of the interesting points from Drogo that you were sharing is you referred to it as the breadbasket and the idea that there's an abundance of food. What do we know from human populations? In my mind, when I'm thinking of those populations that people have studied, where we have very, very long-lived people, I'm thinking of Japan, I'm thinking of Northern Italy, I don't really think of those places as breadbaskets. So what do we know from human populations in terms of this connection between an abundance of food, we're not going to go even into how we're going to measure that, and longevity?
Right. So studies of exceptionally long-lived human populations, the most common concept or the most prolific concept around this are the blue zones. like you mentioned, regions, particularly along the Mediterranean, Japan, Costa Rica, particular communities in Costa Rica, where there's individuals where life expectancy is particularly high, and researchers have tried to say, well, what's so special about these areas? And you're right that it's not about abundance of food, but sometimes certain types of food have been observed to be more common, certain types of diets. A lot has been made about increased reliance on plant versus meat diet, but also rates of activity and lifestyles that are not very sedentary, but instead promote lots of activity. Now, how does that relate to male chimpanzees and their diet? I think an important concept right here, and something I've been talking with some other faculty members about, are the idea of like latent longevity. That, you know, are the Ngogo chimpanzees, could any chimpanzee community become really long-lived if they were given conditions like at Ngogo, or are there underlying different phenotypic differences between Ngogo and other populations? I think Ngogo's proximity to Kaniwara would hint more that many chimpanzee communities could be this long-lived if only they were given available food. But I'm trying to tie it back to thinking about the initial question. I don't feel like I fully answered the question. Let me think.
But I mean, there probably isn't an answer. I was just wondering, because I don't know the literature as well as you do. What were, if people had found a similar kind of possible connection for human populations and food abundance, and you've answered that. I mean, these questions don't have an answer. I was just curious. You know the literature better. Yeah. Thank you so much. I think, Rag, you're next. No, I'm sorry, Clara. Clara, you were first.
Thank you. Clara B. Jones, Social Evolution, retired. Thanks for the talk. I have two clarifying questions and then a question. When you say you have models, do you mean that you have math models?
Yes.
Great. Okay, that's congratulations, by the way. And also, in a lot of primate species, and probably lots of other mammalian species, old individuals are ejected from the group. And in chimpanzees, maybe older individuals could be killed. Is there any evidence of that?
So as far as, they are definitely not rejected. And actually, I, along with a lead co-author, Rachna Reddy, who's a new faculty member at Utah, we actually just submitted an abstract looking at the role of prestige in male chimpanzees, and the idea that maybe these older males are have some additional social status, and some of the evidence for that seems to be that they are particularly valued members. I'm trying not to use anthropomorphic language like the word respected, but that's the way we sort of colloquially look at it, where individuals seem particularly interested in socializing with them. So it's definitely not true that they are rejected in any way. However, being solitary as a chimpanzee comes with risks. At Ngogo, since the study period, I believe 9 of my focal subjects have been killed by other chimpanzees. Some of those are suspected as they were not observed firsthand, but other ones were observed firsthand. Being caught alone by a neighboring community is extremely dangerous. And so being antisocial comes with extremely high costs.
Okay, now my question, related possibly to the terminal investment hypothesis, you find that reproductive capacity does not decrease with age. Is that correct?
As measured by testosterone as a proxy for reproductive investment. Yes. Okay,
well, let's assume that's true. Then we can assume that there is still competition for mates. And there might be strategies to reduce that competition. Now, one thing I didn't see you present when you're talking about feeding data, such as feeding rates, is feeding niche. In other words, a major strategy for reducing competition is to increase the specialization or narrowness of your niche, which could relate to other forms of competition. Do you find any evidence of feeding specialization in older males?
Yes, thank you so much for asking that. So at the Kanyawara community, at the longitudinal study there, they found that older chimpanzees ate less ripe fruit and were more likely to feed on the ground. And so they attributed this to perhaps differences in climbing. I investigated these same patterns, but did not see this. I did not see differences in arborality of foraging behavior or in diet types consumed. I thought that one potential could be mobility, moving up down. Another situation could be avoiding feeding competition, avoiding feeding around other chimps. I think that there's more work to be done to investigate that, but I do not believe I saw any strong evidence or any good evidence for chimpanzees eating differently to avoid competition in particular.
Okay, thanks. I would think more about that issue of competition, though, since they are still in competition with older males. Thanks for the talk.
Yeah, absolutely. Thank you so much.
Thank you, Clara. I think, Rag, you're next, because I don't see any students. Yes, Rag, you're next.
Okay. I am Raghavendra Gadakar from the Indian Institute of Science. Thank you, Ben. That's a really wonderful study population. I study social wasps, so my questions might appear rather naive, or probably naive. I have two questions. One is, I remember reading some papers in the 1990s which found a negative correlation between aging and brain size. And these were interspecific comparisons. And the most sort of popular explanation was that large brain individuals learn survival skills much better than others. My question is, have other people found those kinds of negative correlations since? And also whether in fact one could have a similar pattern in intra-species comparisons. So could some of your chimps which have larger brains survive better, learn survival skills and survive better? So is there anything about brain size that you know of or that you have found? My second question is somewhat more technical. Is there perhaps some confounding factor in your age-related changes in behavior with reference to age-related changes in social status, in dominance or other kinds of social status? Could that be a confounding factor in the age-related changes in behavior?
Thank you so much for the questions. First, regarding brain size and brain size and specifically intraspecies variation in brain size and aging. That's really interesting. That is ringing a bell, but unfortunately, I don't think I know enough about that to comment on at this moment. So I'll put it aside in case I think of anything else. But secondly, the second question regarding confounds of aging, I think that this is one, yes, it is extremely important. And that is one of the reasons I developed the analytical approach I did was this is a cross-sectional study that comes with lots of limitations, but the benefit of it is that we knew these particular chimpanzees and their exact ecological settings very well. And so, I have daily updated ELO scores, for instance, to account for dominance rank throughout the study period. And all of the data presented was collected by myself or Sharifa Namaganda, who's my research assistant at the time, except for the data on dominance, because that is such a long-term project. That is the data where I've accumulated from other members of the Ngoko Chimpanzee Project over many years. And so I feel like in the scope of the data, I think I've done a good job of controlling, incorporating potential confounders of age, such as dominance. in it. However, it's impossible in a cross-sectional study to be completely sure about that. So that is something I think that needs to be looked at. One way to do that is, like I said, the youngest individuals in my study were age 21. And so I was looking, supposedly there we should only see a decline and not a rise. But by looking at young adult males, by looking at adolescents and seeing how the inverse U shape of rank and how that affects things throughout the lifespan, and being able to understand how maybe a low-ranking old male is different from a low-ranking young male, that might help us reveal more about and isolate what are actually effects of senescence as opposed to other things. That's a few thoughts. There was more, but thank you so much for the questions. Is there anything I could further elaborate on?
No, thank you.
Thank you, Rag. Adriana, you're next.
Thank you. Thank you, Ben. I'm Adriana Maldonado. I'm professor at the Universidad de Rosario in Colombia. And I have two questions, and they're mostly conceptual. So the first one, I was wondering, how is senescence defined for male chimps? My understanding is that senescence, at least for females, is defined as a decrease or probably basically the loss of reproductive activity, right? So they cannot reproduce any longer. But testosterone levels, they don't go down, so we can assume that they reproduce, but also you measure copulation rates, and I think they didn't really decrease that much over time. So I'm wondering, these old males, they are reproductively active. So how do we, how comes the senescence part to play a role here? Or is it a different concept, maybe?
No, thank you so much. Some prior research I probably didn't highlight well enough. So the best evidence for senescence, as this really like from a anatomical and physiological perspective, likely comes from body weight. For male chimpanzees, body weight really corresponds to lean muscle mass, and how big you are represents fighting ability. And so when we see declines in that, I think that's one of the most clear and apparent measures of senescence. And we see body weight start to decline in some populations at starting around age 30 for both males and females. However, at sites like Ngogo, it may not start to decline until 35 or even age 40. This is unpublished data. And so reproductive senescence is one really important thing, but there is in particular in females an uncoupling of somatic senescence and reproductive senescence. Something I'm particularly interested in old females is bone health. We see many older females from looking at post-mortem samples. They're extremely osteoporotic. Their bones are incredibly weak, but there's very little idea of, is this a sudden drop off with reproductive cessation, or is this accumulating throughout the lifespan? Other measures such as immune activation measured by urinary neopterin NTX show increased immune activity with age, starting around age 30. That's presented as senescence as well. So senescence is quite like this catch-all term where we can group lots of different things into it, but it is probably worth considering that like reproductive and somatic senescence may be decoupled. It certainly is for females. And it may be in other ways for males as well. I really like thinking about that. Thanks for the question. Is there anything else I could address further?
Yeah, that's great. I mean, it seems that there is some sort of dimorphism in the way males and females get old, right? And it might not be only physiological, but also behavioral. In the case of these males, it seems to be very interesting. Thank you.
Thank you.
Thank you, Adriana. Next question is from, I don't know if you pronounce your name, Laura or Laura?
Laura, yeah, thanks. Yeah, I'm Laura, and I'm a research assistant at Leiden University in the Netherlands at the Comparative Psychology Lab. And thank you for your talk, by the way. And I was wondering, if you could talk a little bit more about this different hypothesis of terminal restriction and terminal investment. And is it sort of a common question to ask, or is it kind of a new hypothesis? Are there animals that do more of the terminal investment and wants to do more terminal restriction? This was, cannot wrap my mind around it completely.
No, thank you so much for the question. So there is terminal investment is pretty well studied and a fairly common framework to look at reproduction at the end of life. And it is typically used a little bit differently than how I was using it, which is important to point out, where it's really seen as not necessarily as like a life stage, but rather what does an animal do when it is sick and it maybe only has a few days left. That's exaggerating on another end of the spectrum. Some of the best evidence, for instance, comes from blue-footed boobies, where they basically, in an experiment, tricked them into oh gosh, I'm forgetting some of the exact mechanisms of it, but they injected them to trick their bodies into thinking that they were very sick. And so the blue-footed boobies who thought they were very sick showed really increased reproductive investment. They were investing more in their young, probably at the end, and other studies have shown that that does come at the expense of long-term lifespan. In primates, there are fewer investigations of terminal investment. There have been investigations of female macaques who do Caio Santiago, and I'm forgetting which particular PIs led that work. But there's not terrific evidence of terminal investment across primates. It is a little less commonly observed. I did look, I mainly looked, I'm like, has anyone used this in chimpanzees before? Because it's a little bit of maybe a strange framework to use in particularly long-lived primates, and it hasn't been very applied. And I don't think I've seen it mentioned in relation to female chimpanzees, but not particularly males. Now, the flip side of that hypothesis, terminal restraint, that there is a lot less evidence on that. And I am a little bit new to this world, but I have not found applications of terminal restraint in primates in particular. So that is a little bit more of a maybe niche idea that I've particularly adopted here.
And the restraint, is it sort of forced restraint, or like they are holding themselves back kind of restraint?
I think it is holding themselves back, but it's also where it is most observed is in older organisms during drought, during dry food seasons, in organisms from insects to birds, where they are going, older individuals are more likely to skip a breeding season if they are in poor physiological condition, or may have some environmental cues that they could expect to be in poor environmental condition. For instance, long-lived albatrosses, this sort of is one of the things to explain. Well, we do see fertility declining with age. We do see number of successful offspring do decline in some long-lived bird species with age. And it looks like those declines might be in some cases shaped as strategically to avoid investing in offspring in a year in which they're unlikely to be successful in the first place.
Okay. Thank you.
Thank you so much.
Thank you. I couldn't help it. I'm looking at the audience, and I'm among the ones who are on the senior side of the spectrum. So this idea that some human beings or chimps, when they are old, they are choosing to restrain themselves, it's only coming, the idea is only being entertained by our younger colleagues. Like if we had a choice, okay, I'm gonna restrain myself with how much exercise I do. Anyway, so Alba, you're next. No, we cannot hear you. Try again. Yep, now I think it's coming. Yep. Now you're muted. Now no, I don't hear you. But for a moment,
no?
there was, now it is. Yes, now it's working. Yep.
Yeah, okay. Hi, Finkel. How are you doing? Good to see you. Well, I'm Alba, University of Buenos Aires, and also a member of the Monkey Project. I have two little questions. The first one is you mentioned, I think it was when you were talking about testosterone, some differences between the West Gogo and the Central Gogo part, and like maybe some explanations that you may have for that, and if you find these differences in other measures as well. And the other one is about the models. You presented this very nicely done visual summary about your models at the beginning. So you have like the outcome variable and then all your controls and mediators and predictors. Did you, maybe I missed some part here, but did you analyze all your outcome variables with the same structure as the models? Okay.
Hi, Alba. So good to see you. So first, testosterone, and I don't think I really emphasize that like testosterone senescence is quite well studied in primates, and so this is clearly like an aberrant. Seeing the absence of a relationship between testosterone and age is weird in this sample and is not consistent with really long-term studies, for instance, at Kaniwara. And so when I say that. When I say I don't observe the difference or I don't observe an effective age, I think that maybe hints at flexible usage of testosterone, not that, oh, there's no testosterone senescence here. Again, I think my study period was particularly weird. They were like, I know so many people are like, they're hoping for, lots of fruit. So the chimpanzees are so super social during their season. And I got that, but I also wanted maybe some rough times for contrast that I did not see. I was in a particularly good year. There are not many investigations of testosterone comparisons between groups because it is much less common to have two chimpanzee communities habituated at the same site. And so I think this is a pretty cool novel data set to look at this. And so the Westerners' testosterone was much higher. And I've done separate investigations to figure out, okay, these two communities, they have different intra-group aggression and competition. They have different numbers of adult males to different numbers of adult females. The West in particular has very few adult males to lots of estrus females, particularly during this period. And they have very different intergroup aggression, where the Westerners throughout my study period were going on patrols constantly. They were ping-ponging around their territory. And when I said that 9 of my subjects died, it was mostly the Westerners killing chimpanzees from Ngogo Central. So this is obviously kind of a cool finding, but I'm relatively new to testosterone, and I'm maybe a little loath to wade into a very socially laden measure, but there is this immediate finding that like, the smaller chimpanzee community that was much more aggressive in intergroup conflict also had much higher testosterone. And from examinations looking at both of those effects and trying to say, was it part about their composition and internal competition, or was it external? It's actually quite weird. Even when I'm controlling for, did they patrol that day? Was there an estrus female? Were they competing? The effective group remains. And so I am confident to say like it is a group level difference. The Westerners are just doing something different than the Centralers during this period. I think that's likely related to territoriality, but it's unclear what. Melissa Emery Thompson, after seeing me, I presented some of that at AAVA last year, and Melissa Emery Thompson sort of had this idea that like maybe they've like hit the ceiling. Like the reason it's not moving and variable that much is because they've like hit some, maybe there's like a physiological limitation on how much you can really upregulate it. And so maybe they're just there, and that's why it doesn't seem to respond that much to the specific patrol events. But that's still only using my data. And for a big something like that has to do with like community events, I want to work with, I'm working with other Ngogo researchers. Jacob Negre, who's at University of Arizona now, just came out with a really interesting paper on testosterone at Ngogo, and it's how it is not correlated with aggression, but is correlated with grooming behavior. So I'll leave it at that, but some really interesting things about testosterone being pretty funky, or to be technical about it, funky, but at least looking different in this community. And your other question was about the model structure. So no, so for a given analysis, so if I'm looking at intake rate, I essentially built lots of different competing models as hypotheses. So, I don't know if I don't want to assume that rank is an important predictor. Rank is positively correlated with intake rate sometimes, but not others. So I built a model with rank and age, and a model just with rank, and a model just with age. And I allow them to compete with AIC. And then if the result says, if AIC is only like, oh, we're 75% sure that both rank and age is good, but there's a 25% chance just age is good, then it's going to weight them so rank will still be in the final model. Yeah. So sorry, not to over explain.
No. It's just like I didn't know, like, because I couldn't imagine how like your hypothesis reflected in your models about like foraging efficiency would be the same for displays or anything like that. So I was like trying to think like how he like would like deal with all these situations. That's fine. Okay, that's all. Thank you, Ben.
Thank you, Alba. The next question comes. By the way, is anybody checking YouTube? We haven't had no questions from YouTube. Thank you, Adriana. Next question is from Ana Maria Romero.
Hello, my name is Ana Maria Romero. I'm currently an undergraduate student at Universidad del Rosario, working with partner preference in guinea pigs. And I had a small question related to if there is some care of the old chimpanzees, because as you explained, they're highly social when there is a high, the fruit temporality, it's high, it's a lot of resources, but thinking about that, they are slow eaters, they don't have a lot of nutrient income, but they also have like this kind of respect that you explained it, Clara about. So I had this question about if there is a care for all chimpanzees, like in a low resource seasons.
Thanks so much for the question. In short, no. There's no social care for them. In talking in introducing this, I usually explain, like, as an elderly human, you can have a bad day. You can have social care in humans is so incredibly important. If an individual is infirm or frail and wants to be brought food, that is important in various human cultures. However, no matter how maybe well liked you are as a chimpanzee or how social you are, no one is going to bring you food. You're still going to have to climb there. There is still food sharing, and we see sharing occur often preferentially along bond strength, but food sharing accounts for a really small portion of calories. It's only certain food sources that are ever shared, like meat, which are not a significant source of calories, at least. And so in short, no, there's no social care. When we talk about them being well liked, it's usually like we affectionately refer to Brownface sometimes as Uncle Brownface, because starting at a certain age, he just seemed to accumulate this sort of gaggle of young chimpanzees who would just follow him everywhere, and he tolerated them, but they weren't necessarily helping him out.
Okay, thank you.
Thank you.
Gracias. Thank you, Ana Maria. Next one, Sari Van Bell. Is it going to be Spanish or English? Sorry.
Well, we can do both. Hello, I'm Sari Van Belle. I'm at the University of Texas in Austin, but I'm actually located in Mexico, and I study the social system of black howler monkeys. I have kind of three thoughts. First one, we have been in the discussion here talking about testosterone, reproductive competition, and so forth of these males. And then so I'm wondering what is there information? I bet there is about paternity patterns actually for these males. So that would be my first question. I don't know if you want my second question already. On top of that, it would be related to space use. Given that you have been talking about these males going on to patrols, if I was an old male chimpanzee, I would kind of figure to stay more central in the home range, and so I'm quite surprised about that, to hear about that.
Yeah, Thank you so much for the questions. So first was, oh my gosh, sorry, we're going. I was thinking about ranging. So I'll answer the second question first, and then you can remind me what the first one was. Participation in patrols does decrease with age after age 40. So older males are more likely to drop out or not join patrols. That's from a 2017 Langer Grabber paper. I would say one of the biggest data sets I have that I haven't even begun to analyze are my GPS tracks when I was following chimps to look at differences in ranging, whether they're being more peripheral in relation to territory. Some has been published on that, but I can't recall the figures off the top of my head about whether they're more or less peripheral. I think there might be some evidence that they actually are more peripheral, because it's sort of surprising, because you're like, that's dangerous, they should know better by that time. But I'm not sure where I don't think very much has been published about where they are and how much they're moving when they're solitary. So would you remind me your first question?
Paternity patterns.
Oh yes. We do know that. It is not something I've analyzed. So yes, so we can talk about reproductive all day, but how successful are these old males actually being? I believe only 5% of the paternities at Ngogo are by individuals over the age of 40. So that is quite small, but I also think when you look at the number of copulations that occur, there are still absolutely more. To me, you can in terms of raw numbers, I actually think it's still a substantial portion. And there was modeling work on chimpanzee longevity in a paper that just came out this year. And they looked at differences in longevity and reproduction. And one of their conclusions was that even just a little bit of reproduction from old males may still be very evolutionary significant. I'm not doing a very good job citing that paper because I've only just seen it, and it came out right as I was finishing my dissertation. So I need to go back and reread that and incorporate it into the study. In terms of understanding particular males and how some of these strategies may impact their reproductive success from an individual standpoint, that's definitely the dream of being able to actually do that. And theoretically, it should be possible through collaborations with other Ngogo researchers, particularly Langer Grabber, as he does the genetics and paternity work at Ngogo.
All right, thank you.
Thank you, Sari. I think I get to ask another question. I'm going to stay on testosterone. And for those of you who didn't join us from the very beginning, I've known Ben for a long time, and we share an interest, a passion, I would say, for trying to get it as best as we could in terms of methods and data. And it was delightful to hear him and Alba discuss how they approach the models in a way that I couldn't do it. So it's great to see my younger colleagues doing it better than I did. But so talking about testosterone in urine, testosterone and the lots of variation we show, and I wrote down here that it didn't decline with age, and then we were engaging on whether we want to make the assumption that it's an indicator. Ben, you rightly said it's a proxy for reproductive function. First, because we were nailed for not having determined with molecular chromatography or spectrophotometry that we are really measuring testosterone. Is it the case? This is a very concrete question. Do you guys know that what you're measuring for whatever is good, it is testosterone?
Yes.
Second, go ahead. No, go ahead.
I could put an asterisk on that, but maybe just me. Yeah.
Because the assays that we use, the commercially produced assays, are very specific, and they're going to bind to one thing or the other depending on what is it that you're actually measuring. The other one, what do we know about the connection in other systems where it's easier between the relationship of testosterone in urine and, say, the changes in receptor functioning of testosterone in the brain with age? Because I don't, how much longer? We know the limitations we have with chimps, with primates, but and I know you feel like me, Ben, because we were talking about this in the morning, how much longer we're going to keep assuming that these proxies are what we want them to be without going and checking in other systems, not to mention captive chimps, the extent to which they are a proxy that gives us this, because it comes down to us, and I know you, Ben, I mean, are we happy with this as a proxy for a refractive function, fertility, or any of that? What can we do?
So I highlighted, as you notice, I sort of cut a lot of results that were age and play a factor out of the presentation, for time, for clarity, for story. But I kept in the no effect of age on C-peptide insulin, which is used as a proxy of energetic status. It should be higher when organisms have more plentiful access to food and lower when diet quality or food access is lower. And I included that because I've had such interesting reactions to presenting that non-result. I first presented this work as really preliminary at a human energetics conference, and I had so many folks coming up to me and saying, I'm also finding no changes in C-peptide insulin. C-peptide insulin is, in my opinion, sometimes I've seen it is sometimes even used as a proxy for fitness, which of course is it is not a proxy for fitness, as it doesn't have anything to do with the number of offspring you have, even though maybe higher ranking individuals in some studies might have higher C-peptide or so there might be some correlations there. But I think overall, what this study shows, I think, or sheds into stark light, is that animals are really good at balancing. And just because you're hungry one day doesn't mean you might have chosen not to eat that day because there's other more important things going on. You can get food the next day. If anything, there was actually the lowest period during my study with respect to C-peptide insulin was when the food was most abundant and when they were going and hunting monkeys every day. So well-established relationships to the highest quality diet period. And that is when, again, it was a very weak correlation, but just numerically, it was the lowest period of C-peptide. And so if you look, so I think that in particular is a measure we need to be really careful of as a proxy for, or assuming, I think we should be careful with C-peptide insulin. I hope I'm not overstating anything. I am not an endocrinologist. I only was able to do all this work because especially during COVID, Melissa Emery Thompson was able to support and do so much of the lab work herself, frankly. Testosterone, at least, like there's so many other systems going on with testosterone. Like you said, there could be receptor, but at least it does seem to me at least that testosterone appears to be honest in various ways, where it is expensive, it is immunosuppressant, and it does have negative effects when it's chronically high. So even if it's doing something different in old males than it is young males, it does still seem like a cost they're paying. So at least I feel more comfortable talking about it like that. And of course, what I've shown is that it is decoupled from reproductive effort. So if you looked at the testosterone and you didn't measure any of the behavior, you would be like, oh, well, they must be copulating just as much or just as aggressive, when that's clearly not true from the behavioral data. So I think it still says something. I think it's still likely saying something about investment and reproduction, but we can't just like measure it and not look at behavior as well.
But no, I know we share on this, but are you aware of studies? I am not, because I'm not an endocrinologist either, but what are the systems where people have compared actually what you get from urine and what you get from, because what if we know the brain is aging, right? We know there are all kinds of changes in the brain associated with age. It could well be that what changes is not the amount of, that it isn't as much how much testosterone you produce that changes, but how your brain takes in the signal that comes out of testosterone. And so we're completely missing the target because it isn't the output of the adrenal gland, but the brain not reading the signal. And somebody must be doing this, if not with chimps or owl monkeys. There has to be some research on this, but I don't know it.
I would love to hear from anyone who thoughts on this, because we've reached. I originally in my drig and my leaky, I originally started out. For the first 2 months of data collection, I was collecting data on both males and females. I didn't sleep. It was exhausting. So I switched to, and so for various reasons I'd be happy to talk about, I switched to just males. And so actually the testosterone was a modification on the original grant. I originally wanted to look at
type 1 collagen NTX to look at bone loss, like I mentioned, but there's much less documented osteoporosis in males. So I thought, well, hey, let's do testosterone. So I'm as I said, I'm a little late to the testosterone game, and I'm learning a lot about this now. So I don't know.
Well, thank you, Ben. It's 1:45. As I announced early, we need to give Ben a little bit of a break before he has his individual meetings. So I want to thank those colleagues who are watching this via the YouTube channel. Remember, you're always welcome to join us via Zoom as well. We look forward to your questions if you're out there. Those of you sticking around with me up until now, thank you again. I look forward to seeing you all on Tuesday. We need to let Ben go, but if some of us want to keep chatting a little bit, I'm up for that. Ben, thank you so much. I don't remember, I think you're meeting Adriana, right? Oh, should we? Well, one possibility is that you guys take a break, and at 1, I'll kick everyone out and you meet here. Let's do that. That's the easiest. Yeah. So I'll be here at 1, make you host, Ben. That way you can keep control of the Zoom meeting, of the Zoom link for the meetings. I'll see you at 1. And to the rest of you, Laura, I don't think we have, thanks for joining us and talking. I think you were at some meetings before, right?
Yeah, a few, something. Yeah.
Good to hear from you.
Eduardo, did you want to turn off the live stream?
Thank you, Adriana.